All right, so we've done glycolysis. We've taken our sugar molecule, we've broken it in half. We've done the oxidation of pyruvate, so our 3-carbon pyruvate. We've actually kicked off one of those carbon atoms as carbon dioxide, and we've linked it up with this thing called the coenzyme A, and that gives us acetyl-CoA, and now we are ready to enter the citric acid cycle. This is where we are going to move from the cytoplasm into the mitochondria. So if we kind of look up here and remember the structure of the mitochondria, we have 2 phospholipid membranes, the outer membrane here and the inner membrane here. The citric acid cycle takes place in the matrix, so on the very interior, so inside of the inner membrane of the matrix of the mitochondria. All right. There's a lot of chemical reactions that happen, and we are just going to hit the highlights that we need to know for this class. So when you guys are looking and studying for looking at other sources, just know that there's a lot more detail that exists out there that you don't need to know. All right. So the citric acid cycle is also called the TCA cycle or the Krebs cycle, just to make it even more confusing than it already is for us. Yay, thanks, science. Okay, so let's talk about these first major steps. So really what we're going to do, we've got our 2 carbons that are coming into the citric acid cycle on that acetyl-CoA. And we are going to bind those with another 4-carbon molecule called oxyl acetate. We don't really have to remember that guy's big scary name, but we do need to remember that it has 4 carbons. Okay, and that when we put that together, it's going to give us a 6-carbon molecule called citrate, also known as citric acid. Okay, and that's called a condensation. So really we're just taking the 2, smashing it together with 4 so that we can start this process. Okay. If we have a lot of ATP already, then we don't need to produce more ATP, so we can actually have this reaction here be turned off if we already have too much ATP. Right, because we remember ATP doesn't last very long in the cells. So if we already have a lot and we make more, then it like doesn't do us any good. All right, so the second step or the second kind of category of what we're going to do is we're going to take that 6-carbon citric acid and we're going to pull off a carbon dioxide molecule, and we're going to get another one of those little electron carriers, and we're going to turn it from this NAD plus into the NADH, and we're putting this in our 88 NADH piggy bank, and we're saving that for later. Okay, so at this step so far, we've produced 1 carbon dioxide molecule. Okay, then we do it again. We're going to pull off another carbon dioxide, and now we're down to a 4-carbon molecule. We're going to make another, transfer some of those electrons from our sugar molecule and put them over onto this carrier. So our NADH, we're just putting that another one in those piles, and we're going to talk about it later. Okay, then we're going to do some rearranging of these molecules. It's going to release some energy so that we can then create an ATP as we rearrange those. The next thing that we're going to do is we're going to, again, do some more rearranging of these 4-carbon sugars. We're going to have another electron carrier, a different kind of electron carrier, FADH2. Always doing, again, just snagging up those electrons. We're saving them for later. We're going to do something with them pretty soon. Okay, and we make another one of these NADHs, and then dun, 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 we are back to oxyl acetate. So now we're ready to have the cycle go again so that we can bring in those 2 carbons from the acetyl-CoA and do the whole cycle again. Okay. So, oops. So some things that we need to remember about this. We do this cycle twice for every glucose molecule. Remember that first step in glycolysis, we took our 6-carbon sugar and split it into 3 pyruvates. So we are going to do this citric acid cycle twice. So we are going to produce 1, 2, 3, 4 carbon dioxide molecules, and we are going to create 1, 2, 3, 4, 5, 6 NADHs and 1, 2 FADH2s and our boy ATP, right? First cycle, 1, second cycle, 2. So altogether, we have 4 carbon dioxide molecules, 6 NADH, 2 FADH2, and 2 ATP molecules at the end of the citric acid cycle.